Sky Island Spice Company.
Mhm.
Local here in Tucson.
Yep.
You're a Tucson native.
Tucson native, born and raised. Yeah.
It's up. It's up. Chad's been here for decades. I think you've been gardening, mushroom expert. I think you've done a bunch of educational talks on
Yep.
seed saving, gardening, and mushrooms.
Yeah. Yeah.
So please give me a warm welcome to Chad. Yes.
Yep. So as he said, I've been a lifelong gardener here in Tucson. I've been working with mushrooms for a little over 10 years. I'm the former vice president of the Arizona Mushroom Society, which is a great organization if you decide that you want to get into mushrooms and foraging and all of the more broad mycological things. I highly recommend the Arizona Mushroom Society just for general information and a good time. So when I started getting interested in mushrooms, I was coming at it from a gardener's perspective. And I started doing a lot of research and, found books this thick, and it was really heavy in the scientific language and a little overwhelming with things to buy, very expensive equipment and all kinds of sterilization needs. And so I did a little bit of experimenting and came up with some other workarounds and just really decided to put this sort of slideshow together to show you various ways you can cultivate mushrooms in our environment without spending tons of money on equipment and just time investment and things like that. You want to click the next slide for me, please? So this is kind of the things I was trying to avoid, which as I got deeper into the rabbit hole of mycology, I did buy some of these things. But, laminar flow hoods that they have in laboratory settings, autoclaves to sterilize equipment, HEPA filters to keep environmental contaminations. Those are things that if you want to have a mushroom farm as a business, you should do that. But as a hobbyist and as a gardener, those things are not very necessary. So next slide, please. So I started to think, what can we do to mimic nature? Mushrooms occur in our natural environment, and the best thing we can do is try to mimic that. So on the right and the left, you'll see oyster mushrooms growing on trees. And then in the very center, I have mushrooms growing in 5-gallon buckets. Those 5-gallon buckets are filled with cellulose-based media. So it could be sawdust, it could be cardboard, it could be a lot of things, which we'll go into a little bit later. But the outer shell of the 5-gallon buckets is acting like the tree bark, kind of protecting the food source from, one, from drying out, and two, from environmental contamination. So that's one way that I recreated what I was seeing in nature by having that barrier. And then there was just a little hole drilled where those mushrooms popped out. That's about a pencil-sized hole for all of those mushrooms to come spilling out. And in nature, what it is when a woodpecker comes along and pecks a little hole, one, they're after the bugs that are inside the tree eating the mushroom mycelium, and they peck a little hole, and then that creates a place for those mushrooms to occur. They also carry that mycelium with them to another tree and deposit it there. So it kind of helps that way. Next slide. So what are mushrooms and what do they do? I'm used to a little bit more interactive group setting. I usually ask the question and wait for a response, but it's on the board. Mushrooms are the reproductive structures of a fungal organism. So when we say mushroom, it's very specifically the reproductive structure of the fungal organism. And so they reproduce via spores primarily. So you see the gills on the undersides of the mushrooms. Some mushrooms have gills and some have pores in various ways that they spread those spores, but most common is the gills. Next slide. So this is kind of the full life cycle of the mushroom. So if you go starting with the spores coming, well, if you look underneath the gills of a mushroom with a microscope, you'll see the organs that produce the spores, the basidia, and those drop spores like crazy, both, we'll say for this purpose, male and female spores, positive and negative spores. So spores are different from pollen. They travel like pollen on the wind, but two spores from the same mushroom can actually share DNA and become fresh mycelium. So spores from the same mushroom can meet, connect with each other, and become hyphae, which then becomes the mycelium. So out of the same mushroom, you can get new mycelium, or it can be spores from different mushrooms of the same species, but it doesn't have to be. It can be from the same mushroom to form new mycelium. Next slide. So here's a great photo of, now this was in an enclosed environment where there was no wind. In nature, this would very rarely occur because the air has to be really still. So the spores have fallen on the edge of the mushroom petal below it and started to become new mycelium, just branching out. And so in nature, that would blow away in the wind and land on some sticks and start to feed on that. But in this case, I got a really cool photo of it just kind of branching out in all directions. So if the environment is right, if it's humid enough, those spores will begin to create new mycelium. Next slide. There's another shot of the same thing happening. You see the 5-gallon buckets there as my container. And then because it was in such an enclosed environment, the mycelium or the spores landed on each other and started becoming new organisms right there on the actual mushroom cap. And it's all over the 5-gallon bucket too. There's white dust everywhere from those spores because they produce millions and millions of spores. But both really good photos of that action happening. Next one. So the fungal organism is made up mostly of mycelium. A lot of times we just kind of colloquially say, I'm growing mushrooms. Really, you're growing mycelium with the hopeful end of getting mushrooms out of it. The organism itself, including the actual mushroom tissue, is made of mycelium. And so here we have mycelium taken from a piece of mushroom tissue that was put on sterilized wheat berries growing, and they're starting to spread onto the wheat berries. This one here was those 5-gallon buckets. When I lifted it up one time, there was nutritive liquid that had dripped out of the bottom, and that mycelium had fully colonized just the liquid on the ground and fruited a mushroom about this, well, cluster of mushrooms about this big. So there wasn't a lot of nutrition for it, but it still was able to eat it all up and then try to reproduce. So that's a really cool thing. And then on the far side there, you can see what we call mycelial primordia. So it's these kind of bumpy nodules that the mycelium begins to make when it's ready to make mushrooms. And so that's usually caused by several things combined. So it's running out of a food source. You know, a lot of plants do that where they're like, survival tactic is make as much seed as possible. Same thing with mushrooms and mycelium. Once it's gotten the sense that it's running low on nutrition, it's going to try to make more mushrooms. It also is by environmental conditions, ideal temperature and ideal humidity. So when all those things are right, or even close to right, it'll start doing this primordia thing. And if those environmental conditions change, if it starts to get too dry, it'll abort and go back within itself, redirect its energy, and then pop out somewhere else later. So if conditions change and it's starting to make a mushroom, it may sometimes pull back and say, oh, it's too dry right now. We're going to try a different time, different place. So that's what the primordia looks like. Next one, please. So you can clone mushrooms from mycelial tissue applied to a food source. With oyster mushrooms, which is primarily what we're talking about today, they eat cellulose and lignin. So pretty much any type of wood or paper pulp is the ideal food source. So you have there, I think that's sawdust. It's growing on sawdust there, and then cardboard. I'll show you guys in a little while a technique of being able to take store-bought mushrooms, putting it on wet cardboard, and then being able to get mushrooms from that store-bought thing. Now, again, this only applies to oyster mushrooms. It doesn't quite apply to porcini or, agaricus, the portobellos and things like that. It, in theory, could, but they have different parameters and they're slower growing, which means you have a higher potential for contamination. The oyster mushrooms are very aggressive growers, and so they can kind of outcompete a lot of other environmental bacteria and molds and things like that. So oyster mushrooms are really the easiest ones to get started with. Next one, please. So here's a great illustration of how you can clone a mushroom from mycelial tissue, and I'll sort of demonstrate that. But really what you want to do is take tissue from the inner part of the mushroom, no matter what type. I like using these, they're called king oyster mushrooms because they're mostly stem, which means there's a lot of inner tissue to work with versus other types of mushrooms where you might not have as much. But you don't want to take tissue from the outside because as it's been handled in the grocery stores and things like that, if there's contamination, it's likely to be on the outside. Mushrooms are very good at protecting their inner tissues from environmental contamination. In fact, they have a lot of antibacterial qualities on their own. And so I like to just peel the outside and then just take a little wedge like that. Then apply it to some wet cardboard. And I'll pass these around for you to see. But in here, I just have a little bit of damp cardboard and we just put it on there, maybe cover it with another piece of cardboard and put it in a little Tupperware like that. And in about 3 to 5 days, the mycelium is going to be not just growing on the cardboard, but actually growing through the cardboard. And I'm going to pass this around for you guys to look at, but I want you to see kind of the bond that the mycelium has with the cardboard after a few days. Let's see if I can pick up this whole thing. So it's all held together, all woven together by the mycelium that is in between these layers of cardboard. And this was about 4 days old here. But I'm gonna pass this around and feel free to kind of pry it open a little bit and see what the mycelium's doing. I'm gonna see if I can get a little close to the camera here. But I don't know if you can see that. But that was growing a little closer. Is that good? Yeah. Yeah, it is. So I'll pass that around and you guys can take a look at it and poke around at it. So it will propagate just on that cardboard. Cardboard is not very prone to having a lot of bacteria and things on it. So you don't really have to prepare it in any special way. We're going to talk about sterilization and pasteurization here in a little bit. But I found that cardboard doesn't carry a lot of contaminants as long as it was dry when you rehydrated it. If it's been out in the yard and it's been rained on and all that stuff, might not be the case. But most cardboard's pretty clean. And then I'm going to show you, I'll pass this around also, but this is one that's been forgotten about for maybe a month and a half or so. And what actually happened is, now if it had the proper oxygen and the proper humidity, it would have actually fruited a little mushroom off of this little tiny bit of cardboard. But what you'll see is that primordia that I was talking about, and it's actually pushing against the lid. It's attempting to pry itself to where there is oxygen. So in nature, it would be coming out of the crack of a tree and it would be putting physical pressure to get itself out of that tree. But I'll show this to the camera here where you can kind of see the mushroom trying to escape there. And also the cardboard is completely covered in white. So I'll pass that around too. This one's a little bit older than the other one that I'm passing around. And so that is really one of the easiest ways you can get started with growing mushrooms at home from a store-bought mushroom. And you could see I really just took a shaving off it. I could probably make 50 mushroom clones with this one mushroom. So That is one easy way to do it. These are king oyster mushrooms. So the question was, what kind of mushroom is it? This technique will work with any oyster mushroom, but these ones with the big fat stems are called king oysters. It's Pleurotus angrii is the species. Mm-hmm. Yeah, so the question was, with the sample that I gave around, was it multiple wedges in between the cardboard? Yeah, there was probably, one every 2 layers or so. Here's another one. I've got like 3 pieces sandwiched between like 5 pieces of cardboard. But you can do it with just 1 piece and 2 pieces of cardboard. It just goes faster. Yes, I would keep them out of direct sunlight, but they not necessarily in the dark. That's a big misnomer about growing mushrooms. A lot of people think that they are grown in the dark, and that's specifically the little white cremini mushrooms that we see in the grocery store. That's the whole reason why they're white. They are genetically the same as portobello mushrooms with the brown tops, but keeping them in the dark makes them white. Most mushrooms actually need a little bit of light to be able to produce, but direct sunlight is not good. So, next slide, please. So this is another technique of cultivating using agar, which is what they use, little petri dishes and all different types of laboratory equipment or laboratory testing. It's just another media. So here I have what they call liquid culture. So it's just a nutritive liquid and the mycelium swimming around in there. And I can take a syringe and stick a bag full of sawdust and grow it from that. That was agar, which is like a little jelly plate. And then the mycelium will grow on that and you can apply it to something else. The reason for growing on agar is if you have like a wild culture or something that you're not sure if it has contamination, like other molds growing on it. If you grow it on agar, it's sort of 2-dimensional, and so it grows rather through it, it grows on it. So if there is contamination, you can cut it out. So agar is a good way of cleaning up your culture, but that's a little bit advanced. But you can see in 24 hours how much it's reaching to jump off onto the food, the mycelium. Next one, please. So what is the mycelium doing as it's spreading out there? It's using a combination of enzymes and physical pressure for the hyphae to penetrate the substrate and break down the polymers in the wood. So it has all these little enzymes on its surface, and anything it comes into contact with, it's starting to digest. As you can see in that little sample that I passed around, the little hairs off of the mycelium are actually penetrating into the cardboard and through it. It's not just coating the outside. So if you ever scraped mold off your food, I'm sorry to tell you, you didn't. But it didn't hurt you anyway. But yeah, so the mycelium, whenever there's mycelium on something, it's actually growing several millimeters or more through it and into it. And in this case, as a wood-loving mushroom, it's digesting the polymers. Lignin is the primary one, and cellulose and all that stuff, and turning them into simple sugars for itself to use. Next one. That's a really cool picture under a micron microscope, seeing the transport of those sugars and things like that. It also, if you have mushroom mycelium substrate that you've grown mushrooms on and you use it in your compost or your garden, it holds moisture really well and helps kind of spread things out. So it's really good in the garden as an add-in after you've grown the mushrooms. Yeah, and so it's expanding the surface area. It spreads all over and through things. It's really unlike any other organism. Next one. So earth soil magicians. Now this doesn't pertain specifically to oyster mushrooms, but it's really cool fact about mushrooms in general. So the honey mushroom is the largest living organism in the world currently. I think it's actually expanded from the time I made this slide, but it is just a massive cluster of mushrooms in Oregon. The honey mushroom, so it grows in the soil, but it's associated with trees. And they've been able to take genetic samples from this side of the forest and miles away, and it's the same genetic mushroom. It's the same organism, and nobody can tell where the mycelium begins and ends. It's the same thing. So that's a really cool thing. It's also this mushroom and several other soil-based mushrooms. They help trees communicate. I've heard it been called the wood wide web, because it's the original internet amongst all kinds of plant and animal life. And they can actually help the trees and other organisms communicate using their network. Next one, please. So, well, that's exactly what I was saying. I knew it was somewhere in there. But yeah, they can help the trees pass on information and divert nutrients. They also share some of those simple sugars with the root systems of trees and things like that, which is very beneficial. And also, as the mycelium becomes tired and starts to degrade and all that stuff, a whole myriad of little beetles and composting insects move in, and they really love that. We'll talk a little bit about that later, because the spent substrate from growing mushrooms is the best worm farm food ever. Like, they love that stuff. Next one. So why would you want to grow mushrooms at home? Well, there's a lot of health benefits to mushrooms. They boost the immune system. They've been found to be anti-cancer in vitro studies, and that's specifically the oyster mushrooms, along with a lot of other mushrooms. They're antiviral. A lot of things that harm our bodies actually harm the mushrooms, so they have natural defenses for it. Fungi are much closer to the animal kingdom than they are the plant kingdom. They breathe oxygen, they produce CO2, they need a lot of the same things. They are susceptible to bacterial infection, and so they have some protections around that. It's also the highest non-animal based protein. Very high in protein. That's true with pretty much all edible mushrooms. However, all mushrooms should be cooked. It's not that raw mushrooms will hurt you, but their protein is tied up in a chitinous material. This mushroom tissue is chitinous, so it's kind of like a seashell or like crab shell. You can't digest it until it's been cooked, and then it softens and you can access the proteins. So if you want the protein out of the mushrooms, always cook them. Otherwise, probably not going to hurt you, but you're not really getting anything other than texture. It's cholesterol free, high in vitamin D and vitamin A, which is all something we need more of, I think. And as a soil amendment, spent substrates add organic matter and structure to the soil. So they kind of create these nice, moist, but not water-heavy pockets. And the composting insects just love it. They live in there and eat it, and really does great things for soil. There's been a lot of businesses in the last 10 years that have come out making mushroom composts and things like that, which I think those are all great things to add, but even better if you can grow your own. There are some mushrooms that have been shown to fight colony collapse, help with the varroa mite issue with honeybees. Specifically, there's what is the name of it? It's a Fomitopsis mushroom. And when they grow on the sides of trees, you see it has this, it looks like dew drops on the bottom of it. And they saw bees visiting and sucking up this dewdrops. And they found that it actually helps their varroa mite infections, taking in some of that. So that's a really fascinating thing about mushrooms in general. Not so much with the oyster mushrooms, but as an ecological side note. Next one. This is my favorite picture here, this is really why I started with mushrooms. As gardeners, we're very much used to the composting process. You take your greens and your browns and you compost it and you get soil. But adding mushrooms into the process, you get a whole protein source, vitamin source. Bioremediation is actually another really interesting thing about mushrooms. They can help clean up the environment. Oyster mushrooms specifically will clean up hydrocarbons. They can eat oil and make it carbon neutral, carbon safe. They did that with the Exxon Valdez oil spill. They did an experiment where they had all this oil-laden material from cleaning up the oil spill. What do you do with it? And somebody had suggested growing mushrooms on it. And the experts at the time said, that'll never work. It's salt water. Mushrooms don't grow on salt. And then we're kind of like, well, has anybody tried it? And nobody had. And it was very successful. And the mushrooms grew to be big dinner plate size. The insect life was healthy on the mushrooms. The bird life was healthy from the insects. And that was a great way of cleaning up that type of mess. On the flip side of that, mushrooms will hyperaccumulate heavy metals. So if it has a lot of lead or mercury or things like that, it does a great job of pulling it out, but you wouldn't want to digest those mushrooms. You wouldn't want to consume those mushrooms. I've heard of a few experiments or theories about using oyster mushroom mycelium up north on the Navajo reservation. They have a lot of uranium mining and things like that, and they have open canals that they irrigate with. And these trucks are going along with their canals. And this was a student group that came up with this hypothesis of, and this was pre-COVID. I don't know if they ever fleshed it out or not, but they thought of creating biofilters in their canals to soak up the uranium dust that may be in their canals before it gets to their fields. And then the part that I really enjoyed, the thinking outside the box, was, well, now you have this, heavy metal toxic stuff. What do you do with it? And they said, well, we should put it in a centrifuge and then sell it back to the mine. So I thought that was a great way of thinking outside the box. And again, I don't know if they were ever successful in experimenting with that, but so it does help clean things up if you use it in a purposeful way, and it can make certain things food safe that wouldn't be otherwise. And then, of course, you get amazing compost. Next one, please. So this is kind of the whole scope. If you wanted to start from spores to growing mushrooms, a lot of times you kind of jump right into the middle here where spawn production and substrate preparation. But you can start with spores and put them on agar and clean them up and do that whole process, but it takes quite a while. So mushroom spawn, as we referred to it, would be this stuff here. So this is just bird seed. Pretty much any type of grain works. And then I've pressure cooked it to sterilize it. Once it's cooled down, then I applied mushroom tissue or other grain spawn that this one's fully colonized here. So you can see it's all white. Put a little bit of that in there, and in a couple weeks, this will look like this. And then I'll put it on its intended food source, which would be a wood-based thing. The reason why we like to use grain as a substrate or a spawn is when we go to spread it onto something, each one of these little seeds is run through with that mycelium. So when you sprinkle them in, there's thousands of little inoculation points versus a piece of cardboard. You have to sit there and tear it into little tiny pieces. And so you just get a lot more spread with something like grain than you would with cardboard. Cardboard totally works, but this works faster. Next one. Next slide. There we go. So you can grow oyster mushrooms on just about anything. I love the whole blue jeans thing, the denim, but all of these things make a halfway decent substrate. Some things are cleaner than others, and some take more preparation, but corn cobs, corn stalks, cotton, all kinds of things, paper products, all that stuff is good for growing on mushrooms. Next one, please. You might have to push it a couple of times, I think, for this. Yeah, keep going. Yeah, one more. There we go. All right, so this is the ideal food source ratios for oyster mushrooms. You want about 70% carbon to about 30% nitrogen-based material. So your carbons would be things like cardboard and paper and corn cobs, things like that. Nitrogen would be things like alfalfa, coffee grounds. There's a whole list of them. But that's ideal. Now, like I mentioned, you can get a mushroom off of just the cardboard, but it's not the ideal food source. It's a little starved for nutrition, but it'll work. But that's the ideal ratio of carbon-based to nitrogen-based materials. Some woods have more nitrogen in them than other woods, and you can science the hell out of it, but that's the basics of it. Next one, please. Here's some ideal carbon sources. So corn cobs, sorghum stalks, wood chips, buffalo grass, and cardboard. That would be a great thing to do with all that buffalo grass. I wonder if it would eat up the seeds. I don't know. Next one. Oh, one more. These are the ideal nitrogen sources. So if you mill your own mesquite, the mesquite chaff still has a lot of nitrogen in it. Any legumes really would. So soybeans, alfalfa, cottonseed or cottonseed meal, coffee grounds, beans, any type of bran. And again, certain wood is a little bit greener wood or younger wood, has more nitrogen in it than plywood or whatever. Those are some great sources for your carbon and your nitrogen. Next one, please. One more. I don't remember why I put that slide in there, but there's some beautiful pictures of mushrooms. Again, it's kind of like going back to the beginning of how they occur in the wild. In this case, it's not a 5-gallon bucket, it's a bag. We call them myco bags. You can get these autoclavable bags that can take high temperatures, and they have a little filter pouch in the top so that oxygen and CO2 can exchange. You can get them on Amazon or any other mycological specialty store will carry those. In that one, I think I have straw and cardboard shavings in there. But next one. So now we're going to talk about how to pasteurize or sterilize your grow media. And there's a few different ways. This is the lowest investment as far as materials or equipment. You're really only investing your time. So it's cold water pasteurization. You're basically using anaerobic bacteria to kill off everything that needs oxygen. And then when you remove it from that environment and expose those anaerobes to oxygen, they die off, leaving it basically sterile. So you can soak it for a couple weeks until it smells terrible and get it out of that water. The problem with this method is, you have this straw or whatever you've soaked, and you have to kind of squeeze the water out of it, and it smells awful, and it gets all over you, and that smell sticks to you. So it takes a while, it smells awful, but there's no electricity needed, there's no chemicals needed. It's just the environment and using those anaerobes to your benefit. I do not recommend but that's one way you can do it. If you have big elbow-long rubber gloves, it might be better. You're well prepared for what you're getting into, which I wasn't. That works, but not the most optimal way of pasteurization. Next one, please. Hydrogen peroxide pasteurization. So you can use a solution of peroxide and water. And I think on the next slide, I might have the ratios. Yeah. So 1 quart of 3% peroxide to a gallon of water to, 10 pounds of substrate, basically scale up until you can cover it. Soak it for 24 hours in that solution and then rinse it. Let the water drain out and then immediately introduce your spawn. With both of these all of these methods, once you take it out, once you've exposed it to oxygen and squeeze all that water out, you don't want to leave it sitting in a room for 2 or 3 days because then other stuff is going to take advantage. So you want to do it pretty quick. It's a pretty safe and easy process. No energy, there's no electricity, no boiling anything. The only expense is the peroxide, which, not terribly expensive. And then that takes 24 hours plus. So faster than the water method, not the fastest. You do have to buy stuff for it, but that also works. The next one. So hot water pasteurization is by far the most common. It's common in our food system. It's common for everything. So it's also the fastest way that you can pasteurize things. So yeah, about 2 to 12 hours. 12 hours is allowing for it to cool down. So that's the trick with hot water pasteurization is if it's still steaming hot, you don't want to put your mushrooms on there because the heat will kill the mushrooms. But once it's cooled down, and especially in sealed jars or those myco bags that I was telling you about, keeps them completely sealed up and waiting for when you're ready for them. So I can sterilize, 30 of these jars and they'll sit around for a week or so until I'm ready to apply something to them. You wouldn't want to let them sit for months, but maybe you could. I don't know. But yeah, it's a way of sterilizing your media. It's relatively easy. I use a pressure cooker, about 2 hours on a pressure cooker will sterilize just about anything. Next. So choosing a spawn. So this is spawn, or really we refer to spawn as anything that you're gonna grow mushrooms from. So the cardboard could be spawn. That's right there in the center. And then grain spawn is the easiest to spread out and easiest to work with in bulk. Next. So these are a couple different types of oyster mushrooms you can grow. These fare better in the warm season, which in our environment is all the time. So temperature ranges from 64 to about 90 degrees. Yeah, and so the mycelium of these will survive in 100 degrees. The mushrooms themselves will not fruit at that time. So your mycelium can get hotter than that and survive it. It's not going to kill it necessarily, but somebody asked earlier about should they be in direct sunlight. Don't put it in direct sunlight, especially if it's in a jar, because it's going to get in there and bounce around and greenhouse effect itself. So indirect lighting is best for growing mushrooms. Next one. So these are a couple of the cool season mushrooms. Again, ideal temperatures, 45 to 70 degrees. Winter mushrooms. Summer mushrooms for other places, winter mushrooms for us. And that's what these king oysters that I showed you earlier are. These are the Pleurotus angrii, and they're all stem. A lot of people like to slice these and make like faux scallops, but they're very tasty and large. Yeah, and then the blue oyster and the pink oyster mushroom, I think, are the most charismatic of the oyster mushrooms. They're very pretty. In my experience, the pink oyster mushrooms do not grow very large. They're very kind of small. They're very pretty, very small. Blue oyster mushrooms will get to be full size, like that. Next one. So these are some parameters for growing your mushrooms: the humidity, the CO2, and the lighting. So humidity at 95 and up percent is too wet. If your environment stays above 95% humidity, it's going to be dripping and it's just too wet. 85 to 90 is fine. Anything above 65 is fine. Below 65, the clusters start to abort. And I'll pass this around. So this was an aborted mushroom that came off the side of one of my blocks. But there's a couple of full-grown mushrooms here. But when you look at the backside, you'll see these little things, little nubs, the primordia that started to make mushrooms, and then it aborted because the humidity was not high enough. I'm gonna see if the camera can see these little knobs where it aborted. Now this is all dried, but those dried out first. So I'll pass that around. Let you see that. So humidity is relatively easy to monitor. You can get a little hydrometer, thermometer, and sort of watch the level of humidity. Now this is the smallest size I would try to grow mushrooms in as a humidity chamber. And so you need to, I only drilled one hole. Really, you should have 2 or 3 holes drilled, 1 or 2 low and 1 or 2 high, because CO2 settles and oxygen comes in from above. So you do need a few little air holes, but to maintain humidity, you can use perlite, the little white stuff that we put in garden soil. Just get plain perlite. You can put it in a pie tin, or you can just put it to one side and hydrate it really well and put that in the bottom, and that will help maintain humidity in your box. But I've found these at the Dollar Tree for $1.25. I'll unplug it here. But it's just like a little diffuser, little oil diffuser that you can get anywhere. But these are $1.25, and it fits perfectly in this little box. And then I just have it connected to an external battery. Now you could plug it into the wall. You could even set it up with a timer. If you have it plugged into a wall, you could have your outlet on a timer where it kicks on for 30 minutes every 3 hours, or, you can dial it in, but you could also just put it on once or twice a day for 20 minutes and it will fog out this whole thing if it had a lid on it. I didn't bring the lid, but if you had a lid on it, it would fog this whole thing out. And those are great for creating and maintaining humidity, because it doesn't just keep one side or one area humid, it kind of sprays it around in the air. So those are great for maintaining humidity. CO2 is a little bit harder to track unless you want to spend a bunch of money on CO2 monitors, but 450 to 550 parts per million, no problem. When you get 700 and up, the mushrooms, if you have mushrooms forming, they'll start to get leggy, just like a seedling that's reaching for light. So they'll stretch out because they're trying to get their head above the CO2 layer that's settled in the bottom. So they'll stretch and stretch, trying to get to where there's better oxygen. So if you see mushrooms that are really stemmy, then that means the CO2 is too high in that environment. So you need to introduce oxygen in some way. And then above that, they get weird and deformed. So lighting, you want dim incandescent lighting or natural lighting, but again, indirect. Yeah, fluorescent lights work great. There was, the U of A did a study several years ago on light spectrum and how that affected how oyster mushrooms grew. And they discovered, I remember it correctly, it was the red light spectrum grew big, like dinner plate size mushrooms, and the blue spectrum grew very small, but a big cluster of very small mushrooms. The weight was the same, the nutrients were the same, everything was the same except for their shape and form. So you get one big mushroom or 20 little coin-sized mushrooms from that difference in light spectrum, which would have some pretty cool implications for the culinary world if you wanted to tailor make your mushrooms one way or another. But yeah, that's just an interesting side note about light. Fruiting temperatures for most of the oyster mushrooms are 65 to 90 degrees. Sorry about that. Next slide. So this is how I have mine set up. It's just a little shelving unit greenhouse. I think it was like 30 bucks. And I can grow probably 10 plus bags of mushrooms or cultures of mushrooms in there. The little white thing in the corner is the big version of this. And then I had it set up on a timer. But yeah, so little greenhouses. We refer to this as a mono tub because you can really only get one thing of mushrooms in there. But that's a very easy way. And I've seen it as easy as taking a trash bag and putting it over your mushrooms and just like spraying the inside of the trash bag with water. So those are ways that you can kind of keep that humidity in while things are drying out or while it's not humid in our environment. CO2, you just kind of have to visually watch for the shape of the mushrooms. They're getting kind of leggy, expose them to a little bit more oxygen without letting them dry out too much. That's the tricky part in our environment. Next one. Those are just some pictures of my mushrooms. So there's some starting out. And actually that one on the far side, I would say that's a sign of a little bit too much CO2. Not terrible, but they're getting a little bit long. So that's kind of what too much CO2 would look like. I think I've only got one or two. Yeah, I think that's the last one there. So these are mushrooms in my compost. There's center one there is my compost that I put into my garden, and it still produces mushrooms. So once you grow your mushrooms, you can harvest once or twice, so you cut mushrooms off, wait another week or so, it may still make more mushrooms. After that, I usually throw it right into my compost bin, and I have blue mushrooms, I have pink mushrooms, these are Phoenix oyster mushrooms, so they're kind of gray. And they just pop up sporadically when conditions are right in my compost pile and in my garden sometimes. That was a couple of years ago. I didn't get any this year, but at least not yet. Maybe this next week if it rains. But I think now I'll open it up for questions. Yes.
So,
Mm-hmm.
if we were to do, oh, sorry. Okay. I guess I'll repeat. I just want to understand the process from all the options you've showed us. So if we were to get what you did and cut a piece of the inner stem of that mushroom,
Mm-hmm.
put it in like the Tupperware,
Yes.
what happens? I think you said after about 5 days, you'll start to see the
Yeah.
mycelium. And then do you transfer it to a chamber like that? Or what do you?
Yes. So, well, what I would do, because this is not very much food for the mushrooms, this is really just getting the culture started. What I would do is, if I was just going to use cardboard as my grow media, right, I would get tons and tons of cardboard boxes and shred them up the best I could, dump them into some hot water, strain it out so it's not dripping, but everything is well hydrated. Put that into a 5-gallon bucket, and then take this that already has the mycelium growing on it and tear it into little pieces And so one other important thing, isopropyl alcohol, you want to clean your hands, your tools, anything that you're really handling the mushrooms with, you should clean with alcohol, so you don't introduce any bacteria. But, I would just take these pieces of cardboard and tear them up like this, and then put them into the bin and maybe do it in layers so you kind of spread it out. That's really why the grain is a slightly better option, because it's easier to spread than tearing all the cardboard. But with this, you have to pressure cook it. The cardboard, you can just get it wet. So kind of different level of intensity going through the whole sterilization of the grain, but the end result is an easier thing to work with at a bigger scale. It has corn in it. This is just like a wild bird seed, but any grain would work. So any grain, millet, sorghum, corn, rye berries, all work well. The main benefit to the grain, one, it's a nutrient powerhouse. So like it has some nitrogen, it has carbon, it has all that stuff. But when you go to spread it into your major grow media, you have all of these inoculation points that break apart really easily, versus the cardboard where you have to kind of work at it, at cutting it up and spreading it around. So that would be the main reason for going with grain over cardboard. But the cardboard is a fun experiment just to see the actual mycelium, growing through the cardboard, so much to where it's actually holding it together, different layers. See, that was cardboard on cardboard, and there's mycelium underneath of it. So all of that is full of mycelium and will carry it on to any other carbon source, basically, that's relatively clean. Now, if you start down this road of putting mushroom tissue on things, and it does it a little bit, but you're not quite sure, and you smell and it smells rotten, throw it out, because bacteria has moved in. Mushrooms should never smell rotten. If they do, that's a sign of bacteria. And then they start to get slimy, but you can always tell by your nose. If it smells rotten, it's no good anymore. Yes, another question.
I have a couple of questions from our Zoom participants. So the first one is, if we have a pine tree stump, would mushrooms work to remove the stump while growing for cultivation? And what plants would you recommend to give shade, and what kind of mushrooms would you plug into the trunk of the tree?
So that's a good question. It would be very challenging in our environment here in Tucson to achieve that, just because it's so hot and so dry much of the year. It's not to say that it couldn't be done. And in other parts of the country, it's very common to do, it's called a log culture, where they take fallen logs and they put wooden dowels that are basically the same thing I did with the cardboard, but they use little wooden dowels, and then they plug that into the tree. It also takes longer to break down intact trunked wood, because the polymers are still holding everything together. In theory, yes, it would slowly break it down, but it would take years. It would take like 5+ years to really do that. And it may even take 2 or 3 years to fruit any mushrooms if you were able to maintain the right conditions. But that's the big challenge here in Tucson with something like a tree stump, is it's very hot and dry and really hard to maintain the humidity around it for the mushrooms to be able to perpetuate. Yes. Okay.
We had a lot of participants tonight.
That's great.
What did you say, what temperature the water should be for the hot water pasteurization?
So for hot water pasteurization, it's really using a pressure cooker. So I think 15 PSI for about 2 hours. But you could also just boil something for 2 hours, as long as it doesn't completely dissolve. It kind of depends on your substrate. So if you had something like wood chips. You could, in theory, just boil those. But really the ideal thing is to pre-soak whatever you're going to grow it on, whether it's sawdust or cardboard or corn cobs. Soak them overnight and then put them into a container. Glass jars are easy, but if you're doing something larger, those myco bags, they're autoclavable bags that you can put through a pressure cooker. So you would hydrate the material, put it in the bag, seal the bag, and then pressure cook that. And so that's how I would go about that.
And one more. What is the best method and media for organic grown mushrooms?
Well, so the best media kind of depends on the mushroom itself. For oyster mushrooms, really any type of tree pulp works. So one common thing is for smokers, they have the little pellets for wood smokers. And that you really don't have to pasteurize or sterilize, because when they make the wood pulp into pellets, there's so much pressure applied that it kind of sterilizes them through the pellet making process. So you can actually take smoker pellets and just hydrate them in a jar and then apply the mushrooms with pretty decent success. Another thing that I've done is, we call it the TAM method. Tam Milton came up with this years ago. This is stuff I learned with the Mushroom Society, and it's using kitty litter, the recycled newspaper kitty litter, not the clay stuff, but it's like little newspaper pellets. Use, 3 cups of that, and then like a 1/4 cup of alfalfa pellets or rabbit food. Put that in there, add water, and then you can go without having to do all of the pasteurization stuff that I talked about. And so that's an easy way that you can just take stuff from a bag, throw it in there, throw the mushroom pieces in there, and go. Yeah. Yes. It's coming.
We've been growing blue oyster mushrooms in a 5-gallon bucket with holes, and we got a couple really nice flushes.
Mhm.
Can we recycle the substrate that's in there now? Just break it and refresh
Yeah.
it with new stuff?
The short answer is yes. And there is a point of diminishing returns in doing that. So if you just take mycelium, tired mycelium that's been eating, and you put it on more food, it will reinvigorate and it will continue to go. But the mushroom production will slowly start to become less and less. And it's not because it's unhealthy, it's actually kind of the opposite. The mushroom learns how to eat more efficiently without having to reproduce. So our goals to get mushrooms out of it is damage, but it's actually getting better at what it does. So that actually will help if you sort of change what food it's eating. That would help keep it on its toes, I guess you could say. So there is a point of diminishing returns if you keep recycling the same mycelium, but I've done it probably 7 or 8 times down the line, and you still get mushrooms. You just don't get as many or as big or as good of production, but you can do that. And so that's the other reason why people do work with spores, is spores kind of prevents that from happening. Same as, take the clone of a clone of a clone. It's kind of the same thing, but it's not evolving with the environment. So seeds offer that chance. Spores, same thing for mushrooms. Yes, I see one way back there.
So do you have any tips about foraging wild mushrooms in the area? Just like what kinds and where, generally, of course.
Yeah. So my first advice is be 100% sure of what you're looking at before you put anything in your mouth. Don't rely on the internet for identification apps that can help you kind of find the right direction, but really the best way is to go with other people who are experienced. So in Arizona, I think we have over 50 edible species of mushrooms, wild mushrooms, most of them being in the mountains. One thing to put your minds at ease is there are no mushrooms, at least in the United States or in the American continents, that is so toxic that you cannot touch it. You will not poison yourself by picking up a poisonous mushroom and then eating something. You won't poison yourself that way. I've even seen people demonstrate with one of the most toxic mushrooms in the United States by chewing it and spitting it out as a method of testing its edibility. I wouldn't recommend doing that, but many people have without any issues. So there's no mushroom that you can't touch or handle or inspect for what it is. With that being said, foraging for wild mushrooms, you definitely want to have people that are very familiar with what you're looking for. There are a lot of mushrooms that will make you sick to your stomach, but won't kill you. The actual amount of deadly mushrooms is very, very small. It's less than 1% in the world.
How do you identify which mushrooms up like on Mount Lemon are good versus those you can't eat?
Well, you really have to be able to identify the exact species of what you're looking at. There's no rule of thumb is like if it's red, it's toxic. If it's white, it's edible. You really just have to know what species. So getting a list of edible species in the area, iNaturalist is a great place to look for, not just for mushrooms, but birds and all kinds of things for wherever you're living or wherever you're visiting. You can look up the flora and fauna. No. The question was, does cooking the mushrooms take out toxins? And it does not. In fact, there are a few poisonous mushrooms that are actually reportedly very tasty. And people don't realize that they've eaten something bad until several hours later. So that is not, there's no real safe way other than knowing what you're looking for. So like these are oyster mushrooms here. They grow usually on fallen pine trees, but they grow on lots of different species of trees. In our area, mostly pine, usually a dead tree that still has its bark. There are a few things that resemble oyster mushrooms, none of them being very toxic. There's one or two that if you taste it, it's spicy in your mouth and you wouldn't want to eat it. But with the oyster mushrooms, it's really the gills that come down into the stem area that kind of set it apart from some of the others. But, there's so many mushrooms out there. There's no one rule of thumb of being a certain color or growing under a certain tree that makes it safe or unsafe. You just really have to know the species. But in the mountains, we have bolete mushrooms, which are like porcinis. We have chanterelle mushrooms. We have oyster mushrooms. We have morel mushrooms. We have all of these things in the mountains. Enoki mushrooms, which are like the long, skinny white ones, only they're not long and skinny in the wild. They're sort of yellow-brown color. And then we call them velvet foot mushrooms. Question? Where do you buy the king oyster mushrooms was the question. So I bought these at G&L Imports over on 22nd and Swan. Most Asian import stores have a good selection of fresh mushrooms, but farmers markets is another place. You could use this technique with lion's mane mushroom also, which is another highly desirable mushroom. It grows a little bit slower, so you got to be a little bit cleaner than you are with oyster mushrooms. But you could also apply that to cardboard and be successful with lion's mane, which is a really delicious mushroom. And you can find it at the farmer's market. Yes.
I have one more question. So my biggest problem with mushrooms is drying out. And I just realized my house is like 74 degrees, so it's already too warm. What is there like a better water to use, or would you just use tap water
So for hydrating,
for hydrating, like for spraying and stuff like
you know,
that?
a lot of times I use tap water. For a long time, I would dechlorinate my water. You know, I'd let it sit for a day,
In a
or I
bucket.
would use distilled water. But in my experiments, it hasn't made a big difference with oyster mushrooms.
Okay.
Other mushrooms may be a little bit more sensitive to the chlorine, but I haven't noticed anything drastic about using the tap water. I have not. I would be hesitant to use rainwater
Was my next question
because
of the
of
bacteria
the
and
vessel
stuff.
that it comes from a tank. I don't know if it's got bacteria.
Yeah.
But it would probably be fine, honestly. But yeah.
Any more questions?
I'm still just a little confused. So I understand we can either do the grain method or the cardboard, like you
Mhm.
showed us, to create kind of the initial inoculation.
Yeah.
And then you said a 5-gallon bucket and you drill a hole or a few holes in the lid.
Yeah.
I guess, can
Well,
you walk us through the whole process?
yeah,
And then
so if
also
I
where
was
the
doing a
box comes in?
Yeah, so if I was doing a 5-gallon bucket, we wouldn't be able to put it in here. So I
Okay.
would have to do something like, you know, a tall Tupperware or something like that. So if I was going to use the box, let's say instead of a 5-gallon bucket, we're doing something this size, right? I would say punch 1 to 2 pencil-sized holes in the side of it. And then while the mycelium is growing on, say you put more cardboard in there, I would tape those holes up to let it kind of grow in there without the holes letting in air to dry it out. If that makes sense. So then once it's all inoculated, once it's covered in white, or if you start seeing the primordia, like the little beginnings on the edges, then you peel that tape off. And that's the point where you would put it in the humidity chamber, the box or the greenhouse or whatever. Because once you expose it to the oxygen, it's going to start seeking a place to go. Like that other one that I was passing around, it was literally trying to pop the lid off so that it could find its way to the oxygen. So once it gets to that stage, that's where you really need to maintain humidity. When it's just in the container and the mycelium is spreading, the container itself can maintain the humidity. But then once it's fully grown and it's looking for a way out, you have to make sure the way out is ideal. If it's too dry, it may start to come out that hole and then shrivel up and die. And then the mycelium goes back into here and regroups, and then it'll start sending an arm this way and that way. And it just tries to find where the optimal place is to make its reproduction. Yeah, so you could use the trash bag method with a 5-gallon bucket. So if you have a 5-gallon bucket and it's got all the cardboard or corn cobs or whatever, and you throw your mushroom pieces in there, like seal it up and wait 2 or 3 weeks for the mycelium to grow. And then once you're ready to do the mushrooms, take the tape off the holes you drilled in the sides of it. And then if you put a big trash bag over it, make sure there's some airspace around it. You can kind of just mist the inside of the trash bag with a spray bottle. And you kind of have to play around with how often you need to do that. Because, you know, October is different than August. You know, August, you might be in there every 10 minutes trying to spray it down. But so you can trap that humidity in with a trash bag just draped over the 5-gallon buckets, and that you can be successful in that way. What I do is it's probably about 5 foot tall shelving unit with a big sleeve over it, you know, greenhouse sleeve. And so I can fit a couple 5-gallon buckets in there. But so that's how I would maintain the humidity for 5-gallon buckets. But you can do it with a trash bag. I've seen people be successful with that. And then it takes usually about a week and a half to 2 weeks once you you peel the tape off and you expose it to oxygen for those mushrooms to come out and become mushrooms.
So you can actually skip that initial step. You can just put pieces of mushroom in there.
You could. The only reason why I would not do that, and I have done it, but there is a potential that the mushroom is carrying some bacteria or some other thing. And so if you throw a bunch of pieces and one of those pieces was carrying bacteria, you just ruined the whole thing. Whereas something like this, it's a little easier to observe. And then the cardboard is less prone to carrying bacteria or other molds, because by the time this is dominating the landscape, so to speak, other things have a hard time moving in on it. But in the transition period, it's a little bit more susceptible. Yeah.
Thank you for this presentation. Just to clarify, do you wet the cardboard first before you put the
Yes.
little sliver on it? Okay.
Yes. Anything that you're going to grow mushrooms on has to be really fully hydrated. You don't want it dripping wet, but you want it fully damp. So with cardboard, it needs to be really flexible and pliable, but not dripping wet.
What about the seeds? Do you add water to the seeds?
Yes. So the seeds, the grain spawn, I soak these overnight. Like I just put a whole bunch of them into a bucket and hydrate them overnight. And then I, dump out the water and actually lay the seeds out on a big towel to dab them free of moisture. Because even with the seeds, if they're too wet, the moisture settles in the bottom and it's not ideal. So you want the seeds actually fully hydrated, but not dripping. And then I put them in the jars. I pack, 10 or 20 of these little jars full of seeds, leave a little bit of space at the top for me to add something. And then I pressure cook them for about 2 hours, and then that sterilizes it. I usually do that at night and then let it sit overnight to cool down. And then the next day I'll apply mushrooms to it, or mycelium. So this is what it looks like once it's fully colonized. And so in theory, like what this is intended for is to apply to wood or cardboard or paper and grow a massive amount of mushrooms. But if I just unscrewed this lid and sat it down in there and covered it up, it would make mushrooms off of the seeds also. So, I mean, it would work as a food source, but the reason why we use the seeds is it's so much easier to break up and spread. So it would kind of be moot. But yeah, but that's what it looks like when it's ready to make mushroom. It should be all white like that. And it's really like one solid mass. Instead of like this, it's still kind of loosey-goosey. Yeah, I usually, I'll take a spoon and wipe it down with alcohol.
Can you
and
repeat the
break it
question,
apart.
Chad? Can you please repeat the question?
Oh, the question was, do you break apart the mycelium or the seeds to spread it? And so I'll usually take a spoon or something that I've sterilized with alcohol and kind of go in there and chop it up because it is so solid. Or if you have good gloved hands, you wipe the gloves down with alcohol, you can just crumble it with your hands. And I would say that other mushroom species are a little bit more delicate and don't like to be handled as much as oyster mushrooms. But oyster mushrooms, like I've literally taken it like jerky and just kind of, pulled pieces like that and dropped it into the thing, and it works. But you're also increasing the chances of contamination doing it that way. I just like to experiment with all kinds of things and see what I can get away with doing. And that's how I found out about the whole cardboard transfer thing is, there's a lot of information on how to grow mushrooms out there. And I was like, do you really have to do all that? Can I just put a piece of mushroom on a piece of paper and make more mushrooms? And I did. And so I figured I should tell people.
And we need to wrap up pretty quick, so maybe one more question, if there's another question.
Well, former life, I was in the culinary arts, and I do cook them at home. I enjoy eating mushrooms, but that's not my primary reason for growing mushrooms. It really came from gardening, and then I discovered this whole other world that I could delve into. With growing mushrooms, what I presented today is like the super basic, easiest way to do it. If you're really into science, like if you're super into hydroponics and doing that sort of thing, mushrooms is a lot like that. You can dial things up and down and make, big dinner plate mushrooms or little itty bitty mushrooms. And you can grow medicinal mushrooms. There's turkey tail mushrooms and reishi mushrooms. There's all these different species with different needs, and learning all of those things is fun for me. But it's not for everybody. I mentioned before so once you get mushrooms off of this and it kind of gets tired, feed it to your worms. I'm telling you, it's the best worm food. They love it.